brain is an interesting organ. It holds our memories, controls emotions, signals movement, tells us when we're hungry, and basically helps us live. But what happens when our brain is not functioning the way it's supposed to? What causes these malfunctions? How do physicians consider these malfunctions in order to diagnose mental disorders? More importantly, what molecular or metabolic pathways do scientists investigate before the physician can make their diagnosis? To examine these questions, let's take a look inside the brain of a schizophrenic. Schizophrenia is a complex, chronic mental health disorder that affects more than 21 million people worldwide. Oftentimes, patients exhibit positive symptoms, behaviors that aren't usually present, such as delusions and hallucinations. They also exhibit negative symptoms, behaviors that are usually there but are missing, such as emotional withdrawal and impaired cognitive ability. On a more scientific level, schizophrenia has hypothesized to result from an excess or deficit of neurotransmitters. For instance, dopamine, the neurotransmitter responsible for reward-motivated behavior. For this reason, some of the more common schizophrenia treatments target the dopamine D2 receptor, such as the drugs chlorpromazine and haloperidol, which have demonstrated clinical efficacy in reducing positive symptoms. Interestingly, people with schizophrenia are more likely to have type 2 diabetes when compared with the general population. A 2 to 5-fold times increase in prevalence of type 2 diabetes has been observed in schizophrenic patients. While the exact cause of this observation is not known and may be due to a number of factors, the increased prevalence of diabetes suggests metabolic involvement as an underlying cause of schizophrenia. Treatment of schizophrenia with antipsychotic drugs has also been seen to cause metabolic changes. In fact, an antipsychotic drug called olanzapine, used to treat schizophrenia, was surprisingly found to also increase the onset of type 2 diabetes and hyperglycemia. If treatment of schizophrenia influences metabolism, perhaps metabolic changes are also related to the cause of the disorder. Although we are very knowledgeable about the symptoms and signs of schizophrenia, the pathophysiology of schizophrenia remains unknown. Based on the observed metabolic changes associated with schizophrenia and its treatment, investigation into alterations in glucose brain metabolism in schizophrenia patients may shed some light on the underlying causes of the disorder. We propose that metabolic changes in the brains of schizophrenia patients result in increased oxidative stress, leading to further changes in metabolic pathways, possibly resulting in the worsening of schizophrenia symptoms. By examining the metabolic changes occurring in the brains of schizophrenics, we can figure out what metabolic pathways are crucial to the understanding of not only schizophrenia, but potentially other mental disorders too. Let's first begin by looking at a study conducted in 2004 by Prabhakaran et al. To examine the first layer of metabolism, glycolysis. Studying transcriptomes, proteins, and metabolites in postmortem brain tissue, abnormal glucose brain metabolism in the schizophrenic brain was examined. It was found that there may be an increased utilization of glycogen stores in the brain, in which glycogen phosphorylase and glucose 6-phosphate transporter 1 were both upregulated in schizophrenia patients. However, enzymes involved in glycogenesis, such as glycogenin and the glycogen branching enzyme, were downregulated. Furthermore, it was also found that the glycolysis pathway was downregulated at the transcript level, in which 7 of the 10 key glycolytic enzymes were significantly decreased in the schizophrenic brain. This included hexokinase, which saw a 1.31-fold decrease, enolase, which had a 1.29-fold decrease, and pyruvate kinase, which had a 1.29 to 1.58-fold decrease, all within the white matter of the prefrontal cortex. These findings suggest that there is an increased demand for glucose and consequently a heavier reliance on glycogenolysis, breaking down glycogen to glucose. Since the enzymes in glycolysis are mostly downregulated, an increased rate of glycogenolysis resulting in the production of glucose 6-phosphate could allow downregulated hexokinase to be bypassed, perhaps acting as a compensatory mechanism to meet the energy demands of the schizophrenic brain. Let's turn our attention to the striatum of the brain. This region is a critical component of motor and reward systems. The striatum receives dopaminergic inputs from external sources and turns them into primary signals in the basal ganglia. In schizophrenic patients, there is an increase of dopamine in the brain. Interestingly, Ganji et al. Found that increased dopamine is associated with increased pyruvate. Studies by Dean et al. Have shown that pyruvate levels in the striatum of schizophrenic brains were 47% higher compared to non-schizophrenic striatum. Following the pathway of pyruvate as the final product of glycolysis, the next step is entry into the mitochondria and conversion to acetyl-CoA. To do this requires the multi-enzyme complex responsible for this conversion, pyruvate dehydrogenase, or PDH. Studies by Dean et al. Showed the levels of the beta subunit of PDH in the striatum of schizophrenic patients were 20% lower compared to non-schizophrenic patients. It can be inferred that perhaps the lower levels of PDH and collective enzyme activity are responsible for the accumulation and increased levels of pyruvate observed in schizophrenic patients. Deficiencies in PDH are associated with 15% increase in lactate production in schizophrenic striatum, which is also due to the accumulation of pyruvate and thus rapid conversion by lactate dehydrogenase to produce lactate. This can also be used to explain the resulting increase in lactic acidosis observed in schizophrenic brains. In the tricarboxylic acid cycle, the TCA cycle, a variety of enzyme expression levels were observed in patients with schizophrenia. First, aconitase, an enzyme responsible for the conversion of citrate to isocitrate, was found to have 18.4% activity, a level which was significantly lower compared to non-schizophrenic control patients. The resulting decrease in isocitrate levels may have caused a chain reaction for the subsequent steps in the TCA cycle, eventually resulting in a significant decrease observed in alpha-ketoglutarate dehydrogenase complex activity, which was also significantly lower than activity observed in the control group. Interestingly, some enzymes in the second half of the TCA cycle were upregulated, which was postulated to be a compensatory response to the reduced activity of the first half of TCA cycle enzymes. A significant increase in succinate dehydrogenase activity, showing 18.3% activity, compared to controls was observed in addition to significant increase in malate dehydrogenase activity, 34% activity. Scientists have postulated that when brain cells are under oxidative stress, succinate will be rapidly oxidized and formed non-enzymatically from alpha-ketoglutarate by transamination in order to shunt the first half of the TCA cycle when alpha-ketoglutarate dehydrogenase is deactivated. This is something commonly seen in schizophrenic patients. When examining all of the downregulated pathways in schizophrenic brain metabolism, Prabhakaran et al. Found that oxidative phosphorylation and ATP synthesis appeared to be the most significantly affected in comparison to control conditions. They found there was a downregulation in the nuclear-encoded subunits of complexes 1, 3, and 4, while their proteomic analysis also revealed significant decreases in complexes 1 and 3. Furthermore, there was also a reduction of transcripts coding for cytochrome C, holocytochrome C synthetase, and ATP synthase complex. Impairment in the ETC, the electron transport chain, can lead the schizophrenic brain to shift its dependence towards anaerobic conditions to compensate for the reduced energy generation, where more lactate would be produced as a result, such as been previously observed in schizophrenia patients. Disorders that involve ETC impairment and lactic acidosis have also been speculated to cause neuropsychiatric conditions such as psychosis. Overall, this reduced expression of some of the key components of the ETC can lead to an increased production in reactive oxygen species, ROSs, and can consequently cause mitochondrial dysfunction. All of the above metabolic changes may have resulted in a redox imbalance in the brains of patients with schizophrenia. This redox imbalance may lead to oxidative stress, which is caused by an overabundance of reactive oxygen species, or free radicals, compared to antioxidants. Reactive oxygen species can result from the decreased expression of the electron transport chain proteins, or from situations in which energy metabolism is altered such as in hypoxia, where a shift to anaerobic respiration may lead to higher ROS levels. Excess ROS can result in damage to proteins, DNA, and white matter. In a study by Kim et al., the balance between the oxidized and reduced forms of nicotinamide adenine dinucleotide, NAD+, and NADH, in the frontal lobe of the brain was measured using in vivo 31-phosphate nuclear magnetic resonance. The redox ratio, the concentration of NAD+ over the concentration of NADH, is indicative of oxidative stress, where a lower redox ratio indicates a low oxidation power and higher oxidative stress. This study looked at the redox ratios of both chronic schizophrenia patients and first episode patients who do not have the additional effects of psychotic medications and compared the results with age-matched healthy controls. 31-phosphate magnetic resonance scanning showed a significant reduction in the NAD+ to NADH ratio in both patients who were chronically ill patients and first episode patients compared to the control group. Both chronic and first episode schizophrenia patients showed an increase in NADH concentration with little to no change in NAD+ concentration when compared with controls, causing the observed reduction in redox ratio. In chronic schizophrenia patients, the redox ratio was reduced by 38%, while in first episode patients, the redox ratio was reduced by 48% compared to controls. These findings demonstrate brain redox dysregulation in patients with schizophrenia, indicative of oxidative stress in the brain. Since NADH production is associated with various metabolic pathways, abnormalities in the redox ratio can be attributed to abnormalities in these pathways, as we have seen. The imbalance of NAD+ and NADH will also have implications for further flux through these pathways, since they play a role in the completion of oxidation and reduction reactions. Further evidence for oxidative stress in the schizophrenic brain was shown in a study conducted by Prabhakaran et al. That looked at the prefrontal cortex postmortem and examined mRNA and protein levels. Results indicated an upregulation of the genes needed to respond to oxidative stress, indicating the increased level of free radicals and oxidative stress in the prefrontal cortex of schizophrenia patients. The protein levels of marker proteins, those that are prone to damage by ROSs, such as aconitase and pyruvate dehydrogenase, were also found to be reduced, furthering the evidence of oxidative stress in schizophrenia patients. Based on these results, schizophrenia may be linked to oxidative stress, possibly as a result of mitochondrial abnormalities or supply of glucose or oxygen. In investigating brain energy utilization in schizophrenia, one study looked at the levels of beta-hydroxybutyrate, a ketone body, in individuals with schizophrenia. Blood samples were taken from 38 schizophrenia patients before and after a 12-week treatment with antipsychotic medication, and serum beta-hydroxybutyrate levels were measured and compared to controls. Levels of ketone bodies in the blood can be used to predict the energy utilization rate of the body. Previous studies have shown that first episode schizophrenia patients display an increased breakdown of membrane phospholipid phosphodiesters, indicating the shift of the brain to using ketone bodies instead of glucose as fuel. As the need for ketone bodies increases, so does the need for fatty acids to fuel ketone body synthesis, resulting in membrane phospholipid breakdown. Before antipsychotic treatment, schizophrenia patients were found to have significantly higher beta-hydroxybutyrate levels compared to controls, 492.3 micromolar versus 327.4 micromolar levels in controls. Following treatment, schizophrenia patients had reduced beta-hydroxybutyrate levels, but levels were still higher compared to controls, now showing a level of 431.5 micromolar. The correlation of beta-hydroxybutyrate levels with the psychopathology and cognitive function of the schizophrenia patients was also measured. Executive function, as measured by a standardized test for cognitive function in schizophrenia, the Brief Assessment of Cognition in Schizophrenia, was seen to decrease with beta-hydroxybutyrate levels. These results suggest that beta-hydroxybutyrate may play a role in protecting cells against oxidative stress, which can lead to a decline in cognitive function. The brain may switch modes to metabolizing ketone bodies as a mechanism of reducing oxidative stress in schizophrenia patients, As such, it has been suggested that a ketogenic diet may be useful in combating schizophrenia. By inducing ketosis, a ketogenic diet may be able to help shift the brain over to ketone body metabolism and supplement the protective effect offered by beta-hydroxybutyrate. From this module, we saw that schizophrenic brains exhibit a decrease in glycolytic enzyme activity and an increase in glycogen breakdown to compensate for this change. Schizophrenic brains also displayed an increase in lactate and decrease in pyruvate flux through the pyruvate dehydrogenase complex. The increase in lactate implies a switch to anaerobic metabolism, indicating hypoxia or decreased mitochondrial capacity within the schizophrenia prefrontal cortex. The downregulation of the first half of the TCA cycle also contributes to the accumulation of pyruvate, lactate, and the reliance on glycolysis for the generation of energy. The shift to reliance on glycolysis in schizophrenia patients is furthered by the downregulation of electron transport chain complexes and their subunits. Taken together, these changes result in an overall increase in the concentrations of NADH, as it cannot be converted back to NAD+ as efficiently by lactate dehydrogenase compared to the electron transport chain. Redox imbalance can lead to oxidative stress through the excess of reactive oxygen species. Oxidative stress can cause further problems in mitochondrial dysfunction and can damage proteins, resulting in further impairments of glucose metabolism. Schizophrenia patients may be able to use ketone bodies to compensate for the reliance on glycolysis and prevent the accumulation of lactate. Since lactate accumulation can lead to the production of ROSs, reducing lactate production by providing an alternate energy source may help decrease oxidative stress in schizophrenia patients. But how does this understanding of schizophrenic brain metabolism help us advance the field of clinical psychiatry? Perhaps the exciting results generated by these studies will impact treatment approaches relating to mental illness, such as drugs to target certain enzymes in aerobic metabolism pathways, promoting their upregulation, or a ketogenic diet to provide additional protection from oxidative stress, benefiting patients suffering from schizophrenia and other disorders alike.